Hi everyone, my name is Nafisa Jadavji. I'm an assistant professor at Midwestern University at the Glendale campus. We have two campuses, one in Illinois and one in Arizona, Glendale. I started my position here in July of 2019. And I'd like to, first of all, before I get started, to thank the executive board of the Journal of Young Investigators for this invitation to speak on this panel on scientific thought and process. It's great to see this conference happening again this year, and kudos to Yug and his team for organizing it. So I'm going to speak to you a little bit about my research that my laboratory does, as well as some of the advocacy work that I've taken, that I've participated in as a scientist, and then how I was involved in the Journal of Young Investigators as an undergraduate, as well as answer some specific questions. So my lab focuses on the brain. I'm a neuroscientist by training. We specifically look at brain health over the span of a lifetime. So from very young to middle age to older individuals. You can see that here in this logo. So we broadly investigate neurovascular mechanisms involved in diseases of aging. We have a specific focus with nutrition and trying to understand the impact of nutrition on changes in neurovasculature. Our focus is specifically on stroke, dementia, as well as the role maternal nutrition plays on offspring neurological health. So we currently have a grant from the American Heart Association where we're investigating how maternal nutrition during pregnancy and lactation impacts stroke outcome in young, middle age, and old offspring. So we use a model system in my lab. Currently, we're working in mice, and so we're inducing stroke in in these mice. You can read more about people that work in my lab as well as our research at my lab website, which is listed down here. So like I mentioned, we focus on nutrition, and our specific focus is on one carbon metabolism, which centers around folates or folic acid, which is well known here for its role in preventing neural tube defects during pregnancy in offspring. So folates are B vitamins. They're water soluble, so we get a lot from our diet. So liver, leafy greens, lentils, breads in the US, as well as other countries, Canada, some parts of or South America have fortified their grains with folic acid in order to reduce neural tube defects. We also make some folate. Our gut bacteria makes some folate, but it's not enough for the daily demands of our body. And so that is why we are able to obtain it from our diet as well as take it in supplemental form from or the chemically synthesized form of folate, which is folic acid. A lot of women that are of pregnancy or of childbearing years will take folic acid. And so like I mentioned, focus on our research is focused on stroke or neurovascular changes, and one change or disease that we see that present in is stroke. And so stroke is one of the leading causes of death in the US as well as worldwide. There are two types of stroke, so there's hemorrhagic and ischemic. In the clinical population, there is a very large proportion of patients that are diagnosed or experience ischemic stroke. With the increased prevalence of, the symptoms associated with stroke, we're seeing that, stroke is turning into this, is resulting into this long-term disability. And we also see so this includes reducing mobility and all of that in survivors, especially old age survivors. This is a graph from it's, I guess now 7 years from the American Heart Association looking at the percent of population that suffers stroke. And so you can see the different age groups here down on the x-axis. And you can see as older individuals, both males and females, there is an increase in the risk of stroke. And so in my laboratory, what we do is we model stroke using, we model ischemic stroke in animals. So we use something called the photothrombosis model, which uses a photosensitive dye. In my lab, we use something called rose bengal and a laser. And what we do is we do an injection of the photosensitive dye, and then we activate that dye using a laser. And we target, so we use a stereotax setup that you can see here, and we target something called the sensory motor cortex. So this is a cortical region in the brain, and what we do is we then are able to measure function both before and after, specifically motor function. And so this is a model of ischemic stroke that we use in the laboratory. I was asked to speak a little bit about in vitro versus in vivo studies, because that's what my lab focuses on. So in vitro work that we focus on is using primary cell cultures. So these are cells that have come from a living organism. We use embryonic tissue from mice. So we grow primary neurons and primary astrocytes in culture, and we expose them to models of stroke, so hypoxia or oxygen glucose deprivation. And we try and understand the mechanisms of how these cells die, how they're impacted by low levels of oxygen and glucose. And so that's why I think in vitro studies are really useful, because you can really get down to the basics and try and understand what's happening. Whereas in vivo studies are a lot more complex because you have a living organism and you have different interactions and things like that. And so I think they both complement each other in the sense that you're learning about basic mechanisms and what cellular pathways are being disrupted or not being disrupted, and then taking that and looking at it in an in vivo, in a living system. It I think is really important. So this is my lab focuses both in vitro, doing in vitro work as well as in vivo work. So this is just a little bit about my research that we do in my lab. During my scientific training, both as a graduate student and as a postdoc, I did a lot of advocacy work. And this is just a summary of some of the papers that I've co-authored. I've done some work looking at the academic job market. So looking at faculty hiring and sort of what are characteristics that are needed to be successful in being hired on as a faculty. So this was published in eLife. We've done some work looking at or trying to develop funding practices for early career researchers that are more inclusive, as well as the importance of writing effective recommendation letters, and then building and sustaining mentor interactions as a mentee. And so I think that's so important in scientific training, is to have mentors and to have this community of support, because being a scientist is quite hard. We face a lot of rejection, a lot of failure in our experiments, grants, writing manuscripts, and so having a support system and some sort of mentors in place is really, really important. Currently, I'm really involved in a initiative called Reproducibility for Everyone. This is looking at establishing reproducible techniques or reproducible ways of thinking in terms of your research and raising awareness of that. And so we have developed several workshops that we give to students, to conference goers. I've taught it at Midwestern. Some of the graduate students I've lectured about reproducibility. And so we've outlined our initiative in a publication in eLife. And so I've really enjoyed all of this work that I've done in advocacy and trying to see, how to improve research culture and being a scientist, especially an early career researcher. It's quite tricky. there's a lot of challenges. And so this has been a lot of fun. It's been challenging. It's been really great to meet very different people. And, I encourage everyone to get involved if you can, in whatever you're passionate about, because then it makes things so much more enjoyable. So leading into that, I was a part of the Journal of Young Investigators as an undergraduate. I worked in the research department as an associate editor and then a research editor, and then I was part of the executive board as a senior research editor. And so I did all of this in my undergraduate and graduate training, and I really enjoyed it. It taught me a lot about the peer review process, providing feedback, learning to be a writer, a scientific writer, which is a lot different in terms of scientific writing versus writing for, English class or anything like that. And then as a PhD student, I was invited back to JYI by the board of directors. So in 2011, I came back, and I've been part of the board of directors now for about 11 years. I served as the vice chair and the chair, and now I'm serving as the finance chair of the board of directors. So JYI has been a part of my life for a very long time, and I've really enjoyed giving back to it and being a part of it. Now I mentor students that run the board or that run the journal, and it's really it's excellent to see how these students grow. And I think it's a really excellent opportunity, even if you're not a science major. We do have a corporate board. And if you're a science major, get involved with the editorial board or the corporate board. It's a great community of people, working towards publication or scientific publication. Yeah, I credit a lot of what I learned as an early career researcher to the Journal of Young Investigators. They kind of gave me that foundation. And yeah, I'm really happy to still be a part of it. So, I guess there are some other topics that I was asked to discuss. The first one was the polarization of science. So we see that happening a lot currently, especially with the pandemic and this distrust of scientists. And I think it's tricky. But I think as scientists, we need to be able to communicate our results to the general public. You're asked, or I've been asked in grants to write lay summaries, so summaries to people that are not uh, um, scientists. And so I have found that to be very useful in developing my skills to be able to communicate to scientists. And I think it's really important as scientists to be able to communicate our results effectively and efficiently to the public. And I think that's one way of reducing this distrust of scientists. I mean, it's obviously a very complex issue, and there isn't one specific way to solve it. Science journalism, I think, is great. I think that being able to communicate results that you make or findings that you make in the lab to the public is really important. A lot of a lot of funding comes from the public, and for that information to be shared with the public, I think is really, really important. Um, we have a great department at the Journal of Young Investigators that does a lot of science journalism, so I think that's really fantastic. And I've tried to do a little bit and share our work through various websites and blogs in order to promote and to share what we have found in the lab. I was also asked to speak about the pandemic effect or the effect of the pandemic on research and all of that. It's been difficult for sure, but I think everyone has had it very difficult. I think the one thing that's been really important is to remain flexible and also to take some time away if you can from work. We have been delayed in some of our experiments, and not as many students have been in the lab. So it's been really tricky. Students have been, doing a lot of stuff virtually. But we've tried to make the best of it, and, turn lemons into lemonade. It has been very tricky, but I think, we've tried to, in my lab at least, focus on everyone staying healthy and, doing the work. And we do a lot of virtual meetings. We have weekly lab meetings and I have weekly meetings with all my students to discuss progress, troubleshooting, things like that. So I think it's important to be flexible, and then also, carve out some time for something fun or just, to have some downtime. And Yug asked me to discuss my favorite memory as a scientist. So my favorite memory as a scientist was when I was an undergraduate and writing a paper. I was in an honors thesis class and we had written a review paper and I wanted to submit it for publication, except I wasn't a great writer as an undergraduate. So one of the professors that taught the class actually sat with me multiple times and we went through the paper to try and make it, ready for publication. I was an undergraduate at the time, so I was quite, young in my scientific career. And what he ended up doing was actually taking my paper and cutting out sentences with scissors and taping sentences together, in a different order than they were already in, so that the introduction at least would flow, and showing me how to create this flow of ideas. And it was a little embarrassing at the moment because we were doing this at the lunchroom table and there was lots of people going in and out of the room. And so I was just a little mortified, but that writing experience taught me a lot about scientific writing. And I actually have part of that work framed and hanging in my office at Midwestern to remind me of how far I've come as a writer, as well as the importance of flow and good scientific writing. So that's all I have for today. It's been great talking about these different topics. Um, if you're interested in contacting me, I am on Twitter. We do have our lab website. And then I'm also available via email if you have any questions or any, if you want some advice or anything like that. I hope you guys enjoy the rest of the conference. And again, I'm really sorry for not being able to attend in person. Um, yeah, great. Thank you so much and take care. Bye.